So we've mentioned the supercoiling of DNA, like we see with our prokaryotic friends. But as we look at a bacteria, being a prokaryote, we know that they lack what? They lack a nucleus, and we really refer to as membrane-bound organelles because the nucleus is considered an organelle. So here we see a bacteria with an electron microscope with a scanning electron on the right. We see that all of this circular DNA is spilled out. And inside, we just refer to that whole thing as the nucleoid. That's going to be sort of this middle region where that circular DNA resides within the bacteria. Now for eukaryotes, we do have the supercoiling, but we go to a different level for packaging and compacting our DNA. Because our DNA is going to be wrapped around a series of proteins that we call histone proteins. And so with our DNA, it's really going to look like a strand of pearls and a pearl necklace. And these are going to be at very regular intervals along our chromosome where we have our histone proteins. Now, when we look at a typical chromosome, we're going to see that DNA, again, averages about 5 centimeters when it is fully unwound and straightened out and lengthened. But once we begin to wind it, we're going to take our histone proteins, and these proteins all together with their DNA piece is referred to as a nucleosome. So we got the histone proteins that are going to have about 200 bases wrapped around them, that whole unit, and what we see here, that's a nucleosome, the proteins and the DNA. So do you see how we're already packaging it? Circling around the proteins, compressing that 200 down instead of a linear length, now we've got it bound around the histones. Now this is a new slide. I sort of found this this morning as I was preparing for the lecture today, because I've always heard if you could unwind all the DNA in the human body, it would reach to the moon. I've always sort of heard that, but I was like, is that really true? So as I looked online and sort of made some calculations, human chromosomes about 5 centimeters. Well, if you take 23 of our chromosomes in each cell, that's 11.5 meters of DNA. Well, you have 2 pair of each chromosome, so you have to double that number. Now, there's debate on how many cells are in the human body, and I found one page that gives a conservative estimate of 10 trillion cells in the human body. So if you take our number per cell times 10 trillion, that comes out to be 1.42 billion miles in length of the DNA in your body attached end to end, end to end to end. And to give you a sense of distance, the moon is only 250,000 miles away from the Earth. The sun is 93 million miles away from the Earth. So not only would your DNA reach to the moon and back, it would reach to the sun and back 7.5 times. That blew my mind. All that DNA is packaged and wrapped up in the nucleus of all your cells. Yeah, that is amazing. So what did people do to try to get at this idea and understand the organization? Well, the first thing they did was they used nucleases, enzymes that just degrade DNA. And what they found was that, okay, once you remove, then you found pieces that differed by 200 base pairs. Remember that 200 base pair in length? That's the nucleosome wrapping. So they're like, that 200 number keeps coming up longer and longer. And if they could remove the protein from the DNA with various organic solvents and proteases, all the DNA was degraded. So that's how they really arrived at our understanding of what we see here as part of our nucleosome. We have a core of 4 histone proteins, and then we have our other histone protein that is situated in the span between. There's about 50 base pairs between our nucleosomes. And so when we take our 4 histone proteins arranged on top, 4 histone proteins on the bottom, those 8 proteins together and our 200, bless you, bases of DNA, that makes up the bead of our pearl necklace. That makes up our nucleosome. And the histone H1 is going to be the linker that sort of ties, you see how it's tying each of those 2 strands together in the twist? So that's packaging the 200, but you're seeing we're shortening the space between each one of our nucleosomes with those linker proteins. So you see how we're beginning to package it? This is only the first level of packaging. It's kind of like proteins in their structure. We have the primary structure, secondary. Well, we're doing the same thing with our packaging of DNA. The first level of packaging being organizing our nucleosomes using the histone proteins. So here's our first level, nucleosomes, pearl necklace. Now the next level is we're going to create fibers. And so now we're going to begin to supercoil our DNA with those nucleosomes, and you see how we're tightening these further and further together. Here, with our linker proteins, we've got about a 10 nanometer height. Here, we've got a 30 nanometer thickness for our fibers. Then, again, because of the supercoiling, we're going to loop our fibers in this manner. So we have these loop domains until finally our loop domains begin to twist on themselves into something that we call heterochromatin. And we're going to distinguish heterochromatin from euchromatin, like in an electron micrographic picture, the light and the dark spots. And then finally, if we take all of these heterochromatin strands, package them together, that's where we get our very highly and most condensed nuclear material, genetic material, in what we refer to as our sister chromatids. There's that X-shaped structure that unfortunately a lot of people call a chromosome, but they're actually identical chromosomes still attached, so we call them sister chromatids. They only become chromosomes once they separate. So here's our heterochromatin, the dark spaces. It's not just in the middle, even the dark area on the periphery, that's heterochromatin. And the light material on the inside is the euchromatin. Don't understand the U because that means good. So good DNA, good true DNA. This typically is the area where we're going to have, it's less condensed so we can undergo transcription, whereas this is much more highly condensed. And this area right in the middle we call the nucleus. This is where we're going to be making a lot of ribosomal proteins, and that's another reason why the nucleus is much darker. So here we've linked our illustrations on the right with actual electron micrographic pictures on the left. There's just a DNA strand. There's our nucleosomes, our fibers. You're going to have to look a lot closer before you're going to see a lot of our loop domains out here in this lighter region. Then here we're getting more and more of our twisted loops. And then finally there is one set of sister chromatids tied together. But that's how we're compacting them down. This we can see with a light microscope. Now again, we've talked about mitochondrial DNA. We've talked about chloroplast DNA. They're really, you can basically relate them to both. We're going to have our circular DNA. We're not going to have proteins. And so once again, because of all that, that gives us the idea that at one point that DNA was more closely related to the prokaryotes than to eukaryotes. Now this is an example of a circular piece of DNA that's beginning to undergo replication. See how we've split that into 2 strands? So when we get into our next chapter and begin to look at that, we'll refer back to pictures that look like that when we begin to look at our better understanding of first prokaryotic DNA replication and then eukaryotic DNA replication. So here's the human mitochondria genome. We have some ribosomal RNA genes, but here we have complex 1, complex 3, and complex 4. Those names mean anything? It's part of the cycle, the very end of our cycle, electron transport chain. So the proteins that are responsible for electron transport in the mitochondria, those genes are actually made by genetic material in the mitochondrial genome. We also have ATP synthase, the gene for ATP synthase. That's in the mitochondria. That's what makes the ATP from the protons flowing through. And so, of course, we're going to see that that's part of the mitochondrial genome. Now, the nuclear envelope. We looked at this thing very early on as we were talking about our organelles. What's a characteristic of our nuclear membrane? Something you can tell me about it. It's porous. It's a double membrane. It's got 2 lipid bilayers, so we've got a double membrane that has pores in it. So it looks like a wiffle ball. That's going to allow material to go in and out. It's also part of what larger organelle system? The ER, and so what do we call that system, do you remember? Endomembrane system. The nucleus, the ER, the Golgi, and the vesicles. So it's all connected and tied in with our endomembrane system. Well, we've already talked about our 2 layers. We've got the porous structure. We can clearly see a nucleus in many of our electron micrographic pictures. And in many times we're going to see that dark spot of heterochromatin that we recognize is going to be the nucleolus. We'll talk more about the nucleolus coming up. So inner and outer membrane with a space between sounds a lot like our mitochondria because it also is a double membrane. But there are a lot of holes in it, and when you look at a mammalian nucleus or even a human nucleus, there may be upwards of 4,000 pores in that nucleus, which blows my mind. It's like, well, the nucleus, is it more whole than nuclear material? Or do you have more stuff? And if you got that many holes, how do you keep anything in or keep anything out? Well, these pores are extremely regulated, and they're guarded by a number of proteins. I mean, in fact, each pore may contain 100 proteins. To me, that sounds like a whole lot of work for a hole. But they're regulatory in nature. They're going to allow small water-soluble molecules, well, they can pass, that makes sense. But larger molecules, such as our RNA, such as ribosomal proteins, transcription factor proteins, these are going to have to be given permission to pass, and they're going to have to be transported into or out of the nucleus. And so as we look at our transport, certainly we already said simple diffusion, water-soluble molecules. Active transport is going to be the principal way that we get a lot of the building blocks and regulatory materials in and out of the nucleus. But the active transport we like to think about is going to be sodium-potassium pump. We've already talked about that pump that we used in our action potential to regain our resting membrane potential from hyperpolarization. Well, that thing comes on when we have a concentration imbalance of our sodium and potassium. But in this case with our active transport, we're going to see it happens as materials need to be transported into the nucleus or out of the nucleus. Now to get in or out, we're going to have GTP. GTP is going to be where we get our energy from. In that third phosphate that's attached, it's going to be hydrolyzed. We recover the energy and use it in our transporter. So let's look on the right. This is our import cycle. So here we have a protein that wants to go in. They call it the cargo protein. It binds to a protein that's called importin. It's not that it's important, but I guess if you want to go in, it's important. Import, in, going in. Think of important as Uber. Okay? Cargo's like, I got to get inside, but I got to have somebody to take me inside so Uber can pick me up. So Uber picks him up. Important comes in, checks in with the transporter. We could think it's the bouncer, or you could think of it's the military guy at the gate before you go into the base. Important and the cargo protein come inside. Then we have GTP added. And once you have GTP coming in with an accessory protein, that releases the cargo to do its thing. And important then is shifted out. Once back out in the cytoplasm, GTP is hydrolyzed, importance free to do the whole thing again. Exporting, kind of the same way. Here we have a protein that needs to come out. Now notice, this is not going to be happening with the same protein. This is happening with a different protein, even though the cycle looks very similar. Make sure you understand that. Because here we have exportin. Exportin's inside. Exportin binds with the cargo protein, with the GTP carrier. That gets exported out. Once it's outside, the GTP is hydrolyzed. The cargo is released in the cytoplasm. Exportin goes back in to do the process again. But notice GTP is in the nucleus, GDP is in the cytoplasm for both. What's the big difference? Whether we're using importin or whether we're using exportin. You're not going to do it without the energy and without those molecules to either go in or out. Everybody see that? See, this is the Uber driver coming in.
So in the export, the cargo
is
released out there. Okay, never mind.
Yeah. See, whether your Uber driver's going in or the Uber driver's coming out, they all have to go in the nucleus to gas up. That's the GTP. They got to gas up to come back out regardless. Right? But exportin is never going to carry anybody in, and importin is never going to carry anybody else out. They're specific for when and how they're going to transport their cargo. So here we are in that nucleus, that dark spot that we saw, the heterochromatin in the middle of our cells. And when we look at, lymphocytes, for instance, when you look at them in histology class, even at the light level, you can see a dark spot inside the nucleus of that cell. And it's one of the defining characteristics, because in addition to that nucleus, you've got heterochromatin around the periphery, so that whole nucleus actually looks like a clock face with numbers around the periphery and the dial right in the middle. So we call it a clock face nucleus. But the nucleus is going to be a place where we have a lot of RNA synthesized, and not just messenger RNA. We have ribosomal RNA, transfer RNA, and we're going to have the ribosomal subunits synthesized, not necessarily packaged together yet. We're going to ship them out. So within the nucleus, we have this nucleus organizing center, specific stretches of DNA that are repeats, so that they can make multiple copies of our ribosomal RNA. And we may, in fact, these repeated units, we may have 5 of these in every human. Do we understand haploid cell? Haploid is half. You got 23 chromosomes. That's going to be your sperm and your egg. And once you become a diploid, you double that number into 10. So all of those nuclear organizing regions are organized into one single nucleus, that dark spot. And it's even darker when you have all the proteins around. So way back when, before we knew so much about DNA structure, restriction enzymes, concatemers, all those big words, how did we really get at inheritable material and how this really works and what the heritable material is? Well, in 1928, there was some work done on pneumoniae, Streptococcus pneumoniae, and mice. Now, when we looked at these, there were different strains. There was a lethal strain and a non-lethal strain. So they said, okay, we've got a smooth and a rough. We know that this, let's see, which are they saying is the smooth one here? So the smooth bacteria, that causes death. The rough bacteria is the non-lethal one. So whenever we looked at that, okay, pretty much as we predicted, but if we boiled pneumonia, if we boiled that bacteria and injected it, the mice survived, whereas when we injected the live, the mice were dead. What do you think happened when we boiled pneumonia? Louder? Denatured the DNA. You denatured the DNA. And also all the proteins that are in there, you denatured those as well. So then they said, okay, well that's an interesting experiment. So if we take the live rough bacteria that don't kill, and we put them with the heat-killed smooth bacteria that didn't kill, you'd expect the mice to be okay, right? No. The mice died. As if somehow these heat-killed bacteria came back to life. Well, that would be one hypothesis, but clearly something else is going on. So then they said, okay, it may not be the bacteria itself. The bacteria may not be coming alive. It might be the DNA. And so if you extracted just the DNA from the smooth bacteria, added that in solution with the live non-lethal bacteria, the mice die. So somehow the DNA was transferred from either the heat-killed bacteria or the DNA that was simply placed in the solution, and these bacteria were able to take it back up, and the living non-lethal bacteria became lethal because of that DNA. So that's one way in which they thought, okay, well, maybe it's the DNA that's doing its dirty work. So they went a little bit further. Here's 1952. Remember the bacteriophages, those little things that we looked at that infect bacteria, the viruses? So they wanted to know, okay, is it the DNA or is it the protein that is really transmitting this genetic information, causing the bacteria to be lethal or non-lethal? So they labeled the protein with a radioisotope called S35, and they labeled the DNA with a different radioisotope called P32. Well, when you took the S35 and you labeled that lethal bacteria and transferred it to the non-lethal, bacteria didn't become radioactive. So they didn't incorporate the protein. But when you look at that lethal DNA that was labeled radioactive, when they placed that material in with the non-lethal bacteria, yeah, the bacteria took it up. So they knew it was something about the bacteria, and with this labeling experiment, they knew absolutely that it was the DNA that had this lethal trait that somehow these non-lethal bacteria were able to incorporate into themselves to become lethal. And that's one way that bacteria do what they do, and that's one way why antibiotics, if you don't take all your antibiotics that you're prescribed, you start feeling better and you stop taking them. Well, there's some bacteria that maybe have mutated that are somehow resistant to that antibiotic. And if you allow them to grow, even when they die, that DNA can be passed on to other bacteria, and now they become resistant, and on and on. You know there are superbugs out there, right? That we have no antibiotics that can get rid of them. All right, so we all know Gregor Mendel, right? The monk that liked to play with pea plants. Okay, he laid down really the foundation of what we know as far as heredity. But he had no clue what genes were. We've got this other guy, Miescher, Friedrich, however you want to pronounce his name. He messed around with white blood cells, salmon sperm, and he discovered nuclein. Okay, now nuclein, it's what he considered at the time to be the genetic material or the hereditary material. And then Zacharias, looking in plant cell chromosomes, he discovered that when you remove the DNA, guess what? The chromosomes disappeared. Now that seems pretty straightforward, right? We know DNA chromosomes. But back in the day, they had no idea. This, I've mentioned before, the DNA fingerprinting. This is how you use restriction enzymes then to get your fragments, and you compare the fragments from one person to the next. And in many cases, when you do compare, let's say in this case, here we have a murder case. We've got the defendant's blood, okay, the person that you think did the killing. You've done some restriction mapping, and you can see these restriction bands, dark one, light, okay. Then, okay, well, there's blood off of the clothes of our victim. Here's our victim's blood, okay. So what would you say? Would you say that the blood on the defendant's clothes, okay, the murderer, where did that blood come from? Yeah, okay, well, he's got that, so I'm saying he, like it wasn't some evil woman. But I mean, they have that one in common, but look at this band. The defendant doesn't have it, but the victim does. There's another band the defendant doesn't have, the victim does. And that's all over. They're close. You think they're innocent. So how did the victim's blood get on your jeans and your shirt?
It's just a coincidence.
Okay, innocent until proven guilty. That's right. Not in the court of Facebook or social media or, on the news. Until what? Found guilty by reasonable doubt.